Okay, it is now my pleasure to introduce our next speaker, Dr. Richie Hodel. Currently, Richie is the Biodiversity Genomics Postdoctoral Fellow at the Smithsonian Institution. His current fellowship aims to improve our understanding of the relative roles of the genome and the environment on shaping polyphenotypic variation on recent and ancient evolutionary scales. In fall 2024, Richie will start as a research scientist holding a position at the University of Michigan. Please join me in welcoming Richie.
Thank you very much, Rebecca. I'm thrilled to be here to share with you some of the work from my Buck Fellowship. And I'll talk to you today about machine learning applications with digitized herbarium specimens and how those can reveal the hybrid origin of the cherry and the plum genus. I think we're all now realizing that the climate crisis is intricately tied up with the simultaneous biodiversity crisis that we're experiencing. Climate's certainly not the only detrimental impact on biodiversity. Other sources like humans driving land use changes are a major threat. But I think the rapidly changing climate is only exacerbating other factors that threaten biodiversity. That brings up the question, what are species' responses to climate change? And if you saw Megan Balk's talk this morning, this is going to be a little bit of a review for you, but I think it's kind of fascinating that plants and animals and all sorts of life on Earth have the same responses to climatic changes. Specifically, species or lineages essentially have a couple options when faced with a rapidly changing climate. They can move, and this means moving to areas with suitable habitat as they're extirpated from their home range, not necessarily seasonal migration or dispersal. This may happen on shorter timescales, such as montane species moving upslope to track their shifting preferred habitat. Or it may happen on deeper timescales, such as lineages moving from one region of the world to another where there's more available niche space or suitable habitat. Another option is for lineages to remain in place, but to develop adaptations to the new climate or environment in their native range. This could include shifts such as plant shifts from having large leaves well suited for a tropical environment to developing relatively smaller ones that are better suited for a temperate environment where freezing events, larger leaves suited for the tropics are not ideal. Finally, the third option is to die. And I mean this in the sense of a species or a lineage going extinct, not necessarily just the death of an individual or even a population. We want to know the components of biodiversity that have moved and or adapted to changing climates. These species and lineages can tell us what traits are important for surviving ongoing and future climatic changes. I want to introduce you to a group that we found has been good at both moving and adapting. And this is the cherry genus, which is also sometimes known as the plum genus. It contains very approximately 250 to 400 species with nearly a global distribution. It occurs in a wide variety of environments, including the tropics, the subtropics, and north temperate regions. Here we see the peach, Prunus persica, as well as a few wild cherry species, Prunus discoidea and Prunus serotina. So as you might imagine, this genus contains cherries and plums, but also some other economically important crop species like apricots, almonds, and peaches. In this genus, historically, there have been three different groups defined by inflorescence morphology, or the arrangement of the flowers. These include the solitary flower group, where we see solitary flowers leading to solitary fruits, such as peach, shown here in green. There's also the corymbose group, which we can think of as paired flowers, so shown here in blue, so think cherries, where paired flowers lead to paired fruits. And finally, there's the racemose group, which has these multi-flowered, indeterminate inflorescences, where we see many flowers growing on a single structure. As you might imagine, growing these solitary or paired flowers is a safer strategy in environments where we're resource limited. These racemes can produce more flowers and then consequently more fruits, but they require much more energy. They're often seen in tropical areas where resources such as water and light are abundant. Both the solitary flower and the corymbose group occupy similar environments. They occur in the temperate zone, where freezing events are common and have a deciduous habit. In contrast, the racemose species are found in both temperate and tropical regions, and they have both deciduous and evergreen representatives. Here we see how some of the racemose species put a lot of energy into producing these reproductive structures, which is easier to do if you're in a tropical environment with abundant resources. You can see the flowers leading to the fruits as you go from top to bottom. However, despite all the economically important species in this group, it's unclear how the major lineages are related to one another. To resolve the evolutionary history of this genus, during my Buck Fellowship, I generated data from 83 species representing all major clades in the genus Prunus. The genomic data I collected include complete chloroplast genomes and 610 nuclear loci, totaling approximately 650,000 base pairs or so of data per species. So why do we want both chloroplast and nuclear DNA? And the answer to that lies within the plant cell. So in plant cells, both the chloroplast and the nucleus have their own genomes with distinct evolutionary histories. Typically, the chloroplast DNA is maternally inherited, whereas the nuclear DNA comes from both parents. So we can think of the chloroplast DNA as analogous to a situation in humans where mitochondrial DNA is maternally inherited, and we can trace it back over our ancestor to mitochondrial Eve. But our nuclear DNA contains information from both our parents because it has those genetic contributions from both parents. So what do we find in the evolutionary relationships? The chloroplast phylogeny showed that the solitary flower and the corymbose groups formed a clade up there on the top in green and blue, and this was all sister to all the racemose species. So essentially, the species were related to one another based on their inflorescence morphology. So what do we find in the nuclear phylogeny? Was the story the same? No, in fact, it was not. And here we see that the solitary flower and corymbose groups still form a clade, but the temperate racemose species are also in this clade. So that's looking at the purple, green, and blue groups up top. This entire top clade is then sister to the tropical racemose group. So essentially, the nuclear data are showing us that these groups are defined by their tropical versus temperate status. So we have different evolutionary trees for the chloroplast and the nuclear data sets, and when we see these differences, it often raises alarm bells and can suggest there's a history of hybridization in the group. So I use an approach called a phylogenetic network to infer if hybridization had occurred. And in contrast to the strictly bifurcating trees like the chloroplast and the nuclear ones we just looked at, where each node is representing an ancestor with exactly 2 descendants coming off of it, a phylogenetic network allows for a hybridization event to occur at some point in the tree. So we used a subset of species to test for hybridization and inferred that one hybridization event in the network was optimal. Here we can see that the temperate racemose group, represented by Prunus serotina, is 71.2% sister to the solitary flower plus the corymbose group, but at the same time, it is 28.8% sister to an early diverging member of the tropical racemose group. So that blue line, that blue diagonal line is indicating the hybridization event. Essentially, we've confirmed the hybrid origin of the temperate racemose group after hypothesizing hybridization based on discordance between those 2 different phylogenies, the nuclear and chloroplast ones. So this history of hybridization can really complicate the inferences we want to make about the evolutionary and biogeographic history of this group. These networks get really computationally intractable really quickly as species are added, so it's considered preferable to use the nuclear phylogeny for these downstream biogeographic inferences where we try to figure out where these species have lived in the past. This is a biogeographic reconstruction of the genus, and I realize it's a pretty complex figure, so please let me walk you through it. This figure is using the nuclear phylogeny, so we see that the temperate racemose species, solitary flower species, and corymbose species form a clade up top, sister to the tropical racemose group. And then if we look at each species listed on this tree, next to them are a small rectangle that indicates where each species lives today. So these rectangles are color-coded based on the continents or the regions that are shown in the upper left on the map. And if you're particularly eagle-eyed, you might notice that some of the rectangles are not colors shown on the map, such as purple. And this is just the biogeographic program getting a little too cute for its own good and trying to show that, for example, a species that lives in both of the Americas, so in the blue and the red area, let's call it purple, but that's kind of confusing, I realize. But that's how it's color-coded. Then if we look at the pie charts that are deeper in the tree at each of the nodes in the past, they show the inferred ancestral regions. Critically, this phylogeny was time calibrated using fossils and molecular clock approaches, so we can see when these divergences occurred in the group, and we can connect these evolutionary events with climatic and environmental conditions in the past. So the hybridization event, if we map it onto this phylogeny, shown in that blue dotted line there, essentially occurred over 50 million years ago and may have provided the genomic novelty that allowed the diversification of the temperate race moose group. One thing that can be really tricky to conceptualize in these biogeographic reconstruction plots is what did the environment look like in the past? For instance, if we focus on that time period between about 50 to 60 million years ago, when the hybridization event might have happened, vast swaths of the northern hemisphere, like where we are today, were much warmer than they are today. So we must consider what the climate looked like in these regions 50 million years ago. And if we look at the plot in the lower left, in that red zone, you can see that the climate globally was much warmer than it is today. It was as high as 25 degrees Celsius compared to maybe 13 or 14 degrees today or so. The climate, in fact, when Prunus was first diversifying around 50 million years ago, looked much more like this. It was much warmer and wetter, a tropical environment throughout areas of the northern hemisphere. Very, very different from today. So why do we care what the climate looked like 50 million years ago? We care because we want to know how these species and lineages responded when the climate changed over the past 50 million years ago until today. In the cherry genus, there were actually two really exciting responses to the changing environment, which makes this a really cool group to study. I'm going to gray out the lower portion of the tree so that we focus on the upper portion for right now. And if we look there, we see that these lineages have an origin in a tropical environment 50 million years ago, in the red box. But then they all transition to occupy temperate environments today. So that's, for instance, the yellow and the teal and the blue region, which is Eastern Asia, Northern Europe, and North America. This is a very rare feat because it is very challenging for species that are well suited for the tropical environments to develop the necessary adaptations to tolerate the much cooler, drier, and seasonal temperate zone where freezing events are common. In fact, this transition has only been observed in fewer than 6% of angiosperm lineages, and the upper portion of the cherry genus shown here is in that 6%. When we consider the ability of species to respond to climate changes, this lineage is very good at adapting. Then if we consider the other half of the tree, we see a very different way of dealing with climatic changes. We know that the conditions were tropical around 50 million years ago, and in the tropical race moose group, we see it move into different tropical regions with bursts of diversification each time. We see lineages diversify in the tropical portions of East Asia, shown in yellow, as well as in tropical regions of South and Central America, shown in red and blue, and also in Southeast Asia, shown in orange, that large lower clade. This is another rare occurrence given that the tropical to temperate transition happened in the top half of the tree. Essentially, this genus has both adapted to changing conditions on top as well as moved in response to changing environmental conditions. Several lineages in the tropical race moose group clearly moved to attract suitable habitat. So using genomic data, we resolved the nuclear phylogeny, revealing that lineages group together based on whether they're tropical or temperate. Moreover, ancient hybridization may have promoted diversification. And finally, as we just saw in the biogeographic reconstruction, this group is very successful at diversifying in response to climatic changes. But this brings up the question, what were the traits or morphological features that were important for the success of this group as the climate changed? I think the fully digitized botany collections at NMNH, millions of specimens, represented a unparalleled opportunity to study the evolution of morphological features. So I want to briefly share with you an approach for using machine learning to classify digitized herbarium sheet images as a way to test phylogenetic hypotheses. On recent time scales, it can be easy to track the morphological features that arise from hybridization, for instance, in this zorse or in this liager. But it's much more difficult when the hybridization took place 50 million years ago. To address this difficulty, I used a machine learning approach with digitized herbarium specimens. Machine learning models really excel when there's lots of data and they can process millions of parameters in parallel. When machine learning models are applied to herbarium sheets, we have ample data from the cherry genus, over 10,000 high-resolution specimens, as well as access to GPUs to process the parameters. Our goal is to then know the morphological features that this group may have acquired as a result of hybridization and determine if this helped it survive environmental change. So let's look at the parents. Machine learning models are really good at classifying images if they have enough data. We already established we have lots of data, so what if we build a model to identify features specific to the two parental groups? Using over 10,000 digitized specimens in the collections, I trained a model that got really good at distinguishing between the cornbow solitary group and the tropical racemose group, or the two parents. The model also used underlying features present in the image data to iteratively improve the algorithm. Then we introduced specimens that were the result of hybridization, the temperate racemose specimens, and we could infer how the model would classify them, and more importantly, why. The temperate racemose species we introduced were nearly evenly classified between cornbow solitary and the tropical racemose parents. But what features were important? We can interrogate the pixels in these images to figure out why the model made a decision. Here we see pixels that had greater importance are shown in warmer colors, like orange, and less importance are shown in purple. For the specimens identified as having tropical racemose features, the racemes were highlighted as important. In contrast, for those that were identified as having cornbow solitary features, the leaf margin and morphology were picked out as key features. Essentially, we see that the ancient hybridization event led to the temperate racemose group acquiring traits needed to tolerate the temperate environment, so smaller leaves, while retaining reproductive structures that were well suited for living in tropical areas, the racemes. In conclusion, I think we've showed that the cherry genus is an outlier in its diversification success. Hybridization clearly introduced genomic novelty, likely spurring diversification, and this machine learning approach can detect both floral and vegetative characters that are associated with the hybridization event. I want to thank a number of people, especially my Buck Fellowship mentors, June Wen and Liz Zimmer, who were very supportive throughout my fellowship and since my fellowship. And I also want to highlight the contributions of Sundari Winslow, who was an undergraduate NHRE intern here, who's here presenting her work at Botany 2023 that was a subset of this project. And she's now in grad school in Finland for plant sciences. And I'm happy to take some questions. Okay, great. Sure, sounds good. That's great. So the question was, why was Prunus persica not included in the study? And so you might remember from early in the talk, Prunus persica is the peach, and so that's clearly a very important member, and clearly there would be a lot of data for it. And the answer gets down to really the pandemic, and I think we had a really good system here where I could go into the lab all by myself, but I couldn't have anyone else there. And I was trying to learn a new method for generating genomic data. And as you might expect, if you're doing it by yourself, and I actually had one of the technicians here, Gabe Johnson, who has infinite patience, talking to me over Zoom and talking me through pipetting a new method. But I had a lot of failures, and Prunus persica was one of those failures, and we ran our first set of data. And we actually know where Prunus should go in this phylogeny because it has a reference genome, but it is not included in that data set because I was clumsy with the pipetter when I was learning early on in my Buck project. Yes. But yeah, it has a full reference genome online, so if you want to download it, you can put it in the tree yourself.
Hey, Richie, thanks for a great talk.
Yes.
I had a more general botany question, and you mentioned that racemose inflorescences were more adaptive for tropical environments, and why is that?
I think just, as you might expect, like indeterminate inflorescences or indeterminate structures like that would, in general, do better if you had more resources around. You might want to put put less resources and less energy into making a single fruit, like a peach, that would be more likely to get consumed and dispersed by something who want to eat the flesh, swallow the stone, walk around, and poop it out and bring it to a new location. A lot of those racemes, though, are structures that you're going to produce a lot more fruits, but they might not necessarily get consumed. A lot of them might end up, rotting on the vine, so to speak. So it's just two different strategies for trying to disperse. And I don't know if it's a 100% universal rule or universal truth, but I think definitely there's a trend in that direction. Yeah.
Cool. Thank you.
Yeah. Thanks.
Thank you, Richie. Amazing use of machine learning. And while Richie was here, as you heard earlier, his mentors were June Wen and now retired curator Liz Zimmer.